All right, so I want to welcome everyone back. This is going to be screencast number 2 for chapter 10. And what we're going to do today is we are going to do our best to help explain the two different processes that occur in photosynthesis. If you remember from our first video, I gave you a very brief introduction to the light reactions. And so what we're going to do is we are going to look at the details of the light reactions, both photosystem 2 and photosystem 1. And then what we're going to do next is we're going to head into the Calvin cycle. And remember, the Calvin cycle is sometimes known as the dark reactions, but I would kind of like you not to refer to it as that, because that can be somewhat inaccurate, and I would like you to refer to it as the Calvin cycle. Now, before we talk about the Calvin cycle, though, as I said, we need to get into some details of the light reactions. And there are two possible routes when it comes down to electron flow in the light reactions. One of them is called cyclic, and the other is called linear. Now, we're going to emphasize the linear electron flow the most in this discussion, but we will briefly talk about cyclic electron flow at the very end of our discussion on linear electron flow. So when we talk about linear electron flow, we are talking about the primary pathway. And this is going to involve, as I had said, both photosystems. So we have photosystem 2 and photosystem 1, and its main job is to produce ATP and NADPH, and we're going to do this by utilizing light energy. Now remember when we had talked a little bit about the photosystem as to what it was. A photosystem is simply a protein, and that protein has special pigments that are embedded within the protein, and these are called chlorophyll. And remember that these pigments are going to basically become excited from a photon, which is a particle of light that's going to strike these pigment molecules. Now remember that we don't have electrons actually being transferred from pigment to pigment. All we simply have is the energy, because those electrons are moving from a ground state to a more excited state. So a photon is going to hit a pigment, and its energy is going to be passed, as I had said, among pigment molecules. It's going to excite eventually a special pigment, and you can see that right down here, called P680. Now remember that P680 simply refers to the wavelength of light that that particular pigment will absorb. So an excited electron from P680 is going to be transferred to something called the primary electron acceptor. Now remember that the anatomy of the photosystem involves not only the protein with the embedded pigments, but it has this sort of light purple area that you see right here. And that's going to be the reaction center of the photosystem. This little square that you see right here, that is going to be the primary electron acceptor. And so we're going to have the removal of an electron from P680, and it's going to be moved to this primary electron acceptor. And what's going to happen with that primary electron acceptor is it's going to pass that electron onto something called the electron transport chain. So before we actually talk about the electron transport chain, we need to sort of have a very, very brief discussion as to what the purpose of water is in these photosystems. So remember that water is going to also be struck by a photon, and that's what's going to split the water molecule. And it's going to be split into two hydrogen protons and some oxygen gas. And there's also going to be the release of 3 electrons from this splitting of water. And so the reason we need to split the water is because when we have the release of that electron from P680, we're going to have an empty spot right here that is missing an electron. And if we don't replace that electron, this process that we're discussing right now is not going to be able to continue. And so one of the 3 electrons that results from this split is going to replace that missing electron so we can have this process be continuous. And as I had said, O2 gas is going to be released as a byproduct of this reaction. So when you think about photosynthesis and you think about the generalized equation of this particular process, remember that oxygen is something that we benefit from. This is where we get our oxygen that we breathe. And so this is going to be the place in photosynthesis where that O2 gas is going to be released. So as each electron enters the primary electron acceptor, eventually it's going to make its way to this area of the thylakoid membrane, and this is going to be called the electron transport chain. So remember that all of this is occurring in the chloroplast, in the thylakoid membrane of that chloroplast. So each electron is going to fall down an electron transport chain from the primary electron acceptor of PS2, and eventually it's going to make its way over to PS1. And so these purple objects that you see right here that are embedded within the thylakoid membrane, These are just the various proteins that this electron is going to pass through as it makes its way down and it bumps through the electron transport chain. So remember, as we're kind of making our way down, the energy that those electrons have is going to become less and less and less. So once it actually makes its way over to P700, you're going to notice that we have another particle of light that's actually going to strike the pigment molecules in photosystem 1. So it strikes the pigment molecules exactly the same way as it had done in photosystem 2. And so the energy that you find here is going to act to sort of energize that electron that has made its way now to this pigment molecule called P700. And so that's going to take that electron and move it from the ground state up to an excited state. So again, it's going to be accepted by this primary electron acceptor within photosystem 1, and then it's going to move to this protein that you see right here. And this protein is important because it's going to function to help produce an electron carrier called NADPH. Now, if you look up here, it says the energy released by the fall is going to drive the creation of a proton gradient across the thylakoid membrane. Now, if you think back to cellular respiration, that proton gradient became very important because we need to build up that gradient to eventually produce the ATP from this process. And so the same thing is happening here. And we're going to do sort of a comparison, a side-by-side comparison of the mitochondria and the chloroplast in just a second to help explain the differences between the two. But the idea is the same. We need that gradient to be able to produce ATP using the same type of enzyme or protein called ATP synthase. So the diffusion of these H plus protons across the membrane, as I had just said, is going to drive ATP synthesis. So as I had said in the previous screen, what you notice here is that that primary electron acceptor that you see right here in photosystem 1 is going to take that electron, and just like it had done in photosystem 2, it's going to fall down that electron transport chain. The difference between the two is this one's relatively short compared to the length that you see right here for photosystem 2. And what it's going to do is it's going to fall to a protein called forredoxin, and that's what this protein is right here. So the electrons are then going to be transferred to NAD+, and that NAD+ is going to be reduced to NADPH. Now what you're going to notice is that we have two very tiny blue dots right here. Both of those blue dots represent electrons. So if you think back to what we had said before, when we had the splitting of water, of the 3 electrons that were released during that split, one of those went to replace the missing electron from P680. We had 2 left over. Those 2 electrons, along with a couple of hydrogens, are going to be taken over to NADPH. Now remember, electrons represent energy. So in addition to the ATP that we're going to produce from this process, we also have the production of NADPH. Both of these products are going to be taken to the next step, which is going to be the Calvin cycle. So you remember up to this point, we've been talking about something called linear electron flow. And I had mentioned that at the very end of our discussion of linear electron flow, I wanted to just briefly mention something called cyclic electron flow. And there's not really a lot of organisms on this planet that actually perform this type of electron flow. It is considered to be relatively primitive and thought to have evolved actually before a linear electron flow. The big thing you want to get from this one is that it only uses photosystem 1. It produces ATP, but it does not produce the electron carrier NADPH. So it's going to generate again surplus ATP. It's going to satisfy the higher demands of ATP because remember, to run the Calvin cycle we need to have some ATP. This type of electron flow is going to satisfy that demand to be able to run that part of the cycle. As I had said, there aren't really too many organisms on this planet that actually carry this out, but one example they do give from your textbook is something called a purple sulfur bacteria. So basically anything that is carrying out basically just cyclic electron flow is going to be sort of a primitive organism of some sort. It's going to be a very simple organism, and in this case it's represented by a bacteria. So the diagram down here towards the bottom is simply what we had just said. Remember that we're not going to have photosystem 2 involved in this one. We're only going to have photosystem 1. We're also not going to have the production of that NADPH that you see over here on the right, and it's going to be cyclic. So we're still producing ATP, but again, we're just producing that ATP to make sure that we can continue the process of photosynthesis by being able to take that ATP and run the Calvin cycle. So as promised, I had said we're going to do a little bit of a comparison between that whole idea of chemiosmosis. Remember, chemiosmosis was where we actually produce the ATP, but what we need to do is we need to compare chemiosmosis in chloroplasts with that of mitochondria. So chloroplasts and mitochondria, again, generate ATP, as I had said, by chemiosmosis, but what they do is they actually use different sources of energy to be able to create that ATP. So if you're a mitochondrion, during cellular respiration, you're going to transfer chemical energy from food. Remember that energy is going to come from the glucose molecule, and you're going to use that to be able to create ATP. A chloroplast, on the other hand, is going to transform light energy, so not chemical energy, but light energy, into the chemical energy of ATP. So that's one difference that you would note in terms of chemiosmosis between the two. Spatial organization of chemiosmosis is going to differ between chloroplast and mitochondrion, but there are also going to be a few similarities that we're going to note. So when they talk about spatial organization, they're just talking about where this process actually takes place. So in mitochondrion, protons are going to be pumped to something called the intermembrane space. So remember we had talked about that in cellular respiration. And that is going to be used to drive ATP synthesis, because what's going to happen is we're going to have a concentration gradient that's been built up. We're going to have more on one side than we do on the other, and that's going to drop back through that special enzyme called ATP synthase. So they're going to diffuse back into something called the mitochondrial matrix. Now again, this is occurring in cellular respiration because we're using mitochondria. When you think about a chloroplast, on the other hand, the protons are being pumped into something called the thylakoid space, and it's going to drive ATP synthesis as they diffuse back into something called the stroma. So again, this was discussing mitochondria. This is discussing the specific parts that are required to pump those protons in a chloroplast. So you got to be aware of the difference between the two organelles. So sometimes it becomes a little bit easier to understand what we were talking about in the previous screen if we sort of had a diagram to be able to help us understand what's happening. So remember, we had two different organelles. One's going to be the mitochondria and one's going to be the chloroplast. Remember, the structures are definitely different. Remember, here we have the mitochondrion that is basically has two primary parts. It has something called the cristae, or these inner membranes that you see sort of folded within the organelle. Then we have something called the matrix. Over here with the chloroplast, remember that we have these sort of flattened discs, which are the thylakoids, and we have this fluid-filled area on the outside, which is called the stroma. Now, when you come down here to this diagram, if you look at the key in the lower left, you have a higher H plus or proton concentration in the dark gray area compared to the lower concentration of H plus or protons that you see with the lighter gray area down here. So if you notice, this is going to be the membrane. So over here when we talk about the mitochondria, this is the inner membrane. When you talk about a chloroplast, this is the thylakoid membrane. We have the same enzyme. This is going to be ATP synthase, so that's not different between the two. They use the same type of enzyme. This is going to be the electron transport chain that we had just talked about when it came down to the light reactions. Remember, we talked about those also in cellular respiration. If you notice, we have a movement of H pluses over to this side, over to the inner membrane space of the mitochondrion, to the thylakoid space when you talk about a chloroplast. These are going to build up in a very high concentration, and once they do that, they're going to naturally fall through this enzyme, because again, we're going to go from concentration that's really high to an area of low concentration. And as they fall through this ATP synthase, remember, we're going to have that phosphate, that inorganic phosphate that's going to be added to ADP. And when you put these two together, that's going to basically encourage the formation of energy, or ATP. Now remember, in chloroplast, this is going to take place in the stroma, but when we talked about the mitochondria in cellular respiration, this was taking place in the matrix of that organelle. So ATP and NADPH are produced on the side facing the stroma, as we had said, where the Calvin cycle is going to take place. So remember, everything that we've talked about up to this point is taking place in the thylakoid membrane. The Calvin cycle, on the other hand, is going to take place in that fluid-filled space. So it's going to take place in the stroma. So in summary, the light reactions to generate the ATP and to increase the potential energy of electrons by moving them from H2O to NADPH is all going to take place within that membrane. And remember, both of these are very important because we need ATP and the energy being carried by this electron transport carrier to be used during the Calvin cycle to be able to run that particular cycle. So what we're going to look at next is we finished up our discussion of the light reactions. Now we need to talk about what some would call the dark reactions. But remember, I told you not to refer to them as the dark reactions. That's a little bit misrepresenting this process. We want to make sure that we refer to it as the Calvin cycle. And this Calvin cycle is going to use ATP, and it's going to use the energy that was contained within NADPH to convert CO2 to sugar. So the Calvin cycle, like the citric acid cycle that we saw in cellular respiration, is going to regenerate its starting materials after molecules enter and leave the cycle. So that's something you're going to notice that's very similar to what we've already looked at back in cellular respiration when we talked about the Krebs or the citric acid cycle. The cycle is going to build sugar from smaller molecules by using ATP, so the energy in ATP, and again the reducing power of the electrons that are going to be carried by NADPH. Carbon dioxide is going to be used to help carbon enter the cycle, and that carbon is going to leave as a sugar named G3P. Sometimes it's called PGAL. So that's glycerol aldehyde 3-phosphate. And that's sort of the precursor to making glucose. So for net synthesis of 1 G3P, the cycle must take place 3 times to be able to fix 3 molecules of CO2 to make this 1 molecule of G3P. Now when you think about the Calvin cycle, there's actually 3 phases that make up this cycle. We have the first one called carbon fixation. Now remember, carbon fixation is simply taking that carbon from CO2 and using it to actually produce glucose. So remember, there's carbon in that glucose molecule. And that's going to be catalyzed or encouraged by a special enzyme called rubisco. The second part is going to be reduction, and the third part is going to be the regeneration of the CO2 acceptor called RuBP or RuBip is what it's referred to sometimes. Now we'll look at these as we make our way into the diagram. Alright, so this is the diagram that I was referring to from the previous screen. So if we start way up here at the top, you're going to notice that we have the input of a CO2 molecule into the Calvin cycle, and it's going to enter into it one at a time. And so as it enters into the cycle, you're going to notice here's that special enzyme that we had referred to called rubisco. Now that rubisco is going to take that carbon that's on that CO2 and it's going to add it to this molecule that you see over here on the left called RuBip. And so that is going to cause a very short-lived intermediate molecule, a 6-carbon intermediate, to be formed. When you form that intermediate, it's going to break down super quickly. Now, before I go on to the next part of the Calvin cycle, remember there were 3 phases, and this one's going to be discussing carbon fixation. So remember, fixation was simply taking that C from that carbon and adding it to another molecule. So as I had said, that short-lived intermediate is not going to hang around too long. It's going to eventually form 3 phosphoglycerates. And so remember, we had a 6-carbon molecule here, and this is going to be broken in half. Now, what I'm going to have you do also is, I'm not going to have you worry too much about the numbers that you see here. If you notice, we have a 3, a 6. Eventually, that kind of plays into the idea of producing the sugar that you see down here at the bottom. But for all intents and purposes, you need to not worry too much about that. So down here, if you notice, we have 3 phosphoglycerate, and that phosphoglycerate is going to basically utilize the energy that we have from ATP. Remember, that energy came from the light reactions. It's going to take that ATP, it's going to basically break it down into ADP. It's going to release that phosphate, and that's going to attach the phosphate to this next molecule that you see called 1,3-bisphosphoglycerate. And so now we have 2 phosphates instead of just 1. Then if you notice, we have the energy from the NADPH. It's going to be oxidized, and it's going to be bumped back to NAD+. And if you notice, we also have some phosphates that are here as well. And that's going to basically be considered the reduction phase of the Calvin cycle. And if you notice, the next molecule we're going to form is something called glyceraldehyde 3-phosphate. So that was the G3P that we had mentioned before. Now this is a really super important molecule because if you notice, this is going to be the part of the cycle where you actually get the production of sugar. And so if you notice, G3P, a sugar, this is sort of like the precursor to actually making that 6-carbon sugar. So glucose and other organic compounds are going to be formed from this process. So then if you notice, what we have to do here is we're only going to make really 1 sugar molecule. We're going to have some excess, actually, G3P that's going to be present down here in this part of the cycle. And the reason we have some extra G3P is because we need to bump that over to the regeneration phase of the Calvin cycle. And so if you notice, that's going to be moved over here to the left, and we're going to use a little bit more of that ATP, and it's going to change that G3P basically into RuBip. Now remember, we had talked about that RuBP. This is going to be a 5-carbon structure, but remember, we're going to change it into that short-lived intermediate by actually taking the carbon from the carbon dioxide that enters the system and adding it to this molecule. So that kind of demonstrates that this whole cycle is going to be hopefully continuous during photosynthesis. And remember, the primary purpose of the Calvin cycle is to take that CO2 to fix it and to help us produce the food for the plant. Remember, it's going to be used to produce the sugar as a byproduct of this process. So what we've done so far is we've actually talked about what we consider the most common way to fix carbon on this planet, but there have been some alternative mechanisms that have actually developed over a period of time, and most of those have developed or evolved because there are some plants out there that actually have the ability to live in very hot, very arid climates. And so the problem with this is you get something called dehydration occur. And when you have dehydration, the stomata, which are the very tiny holes that you find in the leaves of plants, these tiny holes actually allow CO2 to come in and O2 to leave. But what the plant's going to do is, if a plant is exposed to very dry conditions, it's going to do its best to try to protect itself. And so on hot, dry days, the plants are going to close their stomata. And what that does for the plant is it actually helps them to conserve that very important water. But the problem is it also limits photosynthesis. So the closing of the stomata is going to reduce access to CO2, because remember that CO2 can no longer get into the plant through those stomata from the environment. And the other thing that's going to happen is it's going to cause a buildup of O2 within the plant as well, because CO2 and O2 is actually passing back and forth through those stomata. So these conditions are going to favor something called photorespiration. And what we do is we categorize plants into 3 categories. The first category is something called C3 plants, and those are the ones that we've been talking about up to this point. So those are the most common group of plants. Initial fixation of CO2, again via rubisco, remember the enzyme that we had talked about in the Calvin cycle, is going to form that 3-carbon compound. So that's going to be the G3P that we had talked about, that precursor to the formation of glucose. In photorespiration, rubisco is going to add O2 instead of CO2 in the Calvin cycle. So remember we had talked about that CO2 being fixed at the very beginning of the cycle. Remember we changed that 5-carbon compound into a 6-carbon compound using the C from carbon dioxide. The problem is rubisco really doesn't care whether or not it adds a C or an O, whether it uses carbon dioxide or oxygen. And if we have conditions where there's no CO2 coming in, so we don't have a really high concentration of CO2, it doesn't make a difference. It's just going to go ahead and add it's going to add the O2 instead of the CO2. And what that does is it consumes O2 and organic fuel releases CO2, and the unfortunate thing with this process is it does not produce ATP and it does not produce sugar. And if you can't produce ATP and you can't produce sugar, then what's going to happen is we're going to have a hard time carrying out the Calvin cycle. So in many plants, photorespiration is a problem when you have hot, dry conditions because it can drain as much as 50% of the carbon fixed by the Calvin cycle. So as we had said, there are some alternatives to this idea of living in a very hot, dry climate, because we do have plants that live in those climates. One alternative is where we have a group of plants called the C4 plants that are going to minimize the cost of photorespiration by actually incorporating CO2 into 4-carbon compounds in the mesophyll cells of the plant. Now this step is going to require the enzyme PEP carboxylase. And so what's going to happen is PEP carboxylase actually has a higher affinity for CO2. So it's different from rubisco. Remember we said rubisco really doesn't care whether or not it fixes CO2 or uses the O2. And remember, if it uses the O2, it sort of halts the whole Calvin cycle. PEP, on the other hand, it only wants the CO2. So it can fix CO2 even when CO2 concentrations are very low. So even if there's not a lot of CO2 in these cells, it can still go after them and actually fix the carbon dioxide. These 4 carbon compounds are exported to another type of cell called the bundle sheath cells, and this is where they will actually release the CO2, which then can be used in the Calvin cycle. So over here on the right, what we have is we simply have sort of a description of the leaf, and this is just going to reference the different cells that I was referring to as we discussed the C4 plants. The mesophyll cells are going to be these cells that you see right through here, and the bundle sheath cells lie very close to these mesohyl cells. And over here on the far right, this is simply the pathway in a diagram form. So remember, we had said that CO2 that is still there. Remember, we have a plant here that is sort of kind of CO2 deficient, but there's still some there. Instead of using Rubisco, we're going to use PEP carboxylase, and that's going to have a really high affinity for CO2. And so that PEP carboxylase is going to take that CO2 and form something called oxoalloacetate. And that particular chemical is going to be forming something called malate. Now once it forms the malate, you're going to notice it's going to break into 2 components, one of those being pyruvate, and that pyruvate is something we had talked about in cellular respiration, and then we have one called CO2. And so this is actually acting to help sort of increase the concentration of CO2 within the plant cells. And so if you notice, we've transitioned from the mesophyll cells to the bundle sheath cells, and that's where the Calvin cycle is taking place. So the Calvin cycle is going to go ahead and basically grab on to that CO2 that's being produced and go ahead and run that Calvin cycle normally and produce the sugar that the plant needs for food. The pyruvate, on the other hand, is going to basically help to regenerate this process and basically be used to create more of that PEP molecule that you see right here. And again, if you notice, we have a little bit of ATP that's going to be used to be able to do that. So again, this is an alternative to the C3 plants, which have a really hard time tolerating these very dry and hot conditions because, again, they're sort of compromised because they close those stomata and there's not as much CO2 coming into the cell. Now, the very last category that we're going to look at is one that, again, is another alternative to the C3 plants. There are some plants that are categorized as CAM plants, and what they do is they actually use something called crassylation acid metabolism, and that's what CAM stands for. And they're using that to, again, fix carbon. CAM plants, what they're going to do is they're simply going to open their stomata only at night. So once daylight hits, they're going to make sure that those stomata are closed really tight. And the reason they do that is because, remember, they don't want to lose water. And so these would be the type of plants that you would think about that are called succulents. And a succulent would be something like a cactus or maybe a jade plant. And so what they're going to do is they're going to incorporate CO2 into organic acids. So the stomata are going to close during the day, the CO2 is going to be released from these organic acids, and it's going to be used in the Calvin cycle. So down here towards the bottom, this is just a really quick representation of a CAM plant and what it's basically doing chemically within the leaves of the plant. And so this is going to be the mesophyll cells that you would find in the plant. And if you notice, we have CO2 coming in, so we still have the CO2 coming in during the nighttime, and it's going to form a product called malate, and that malate is a 4-carbon compound, and that malate is going to be used to create, again, 2 byproducts. It's going to be used to create pyruvate, which again is a 3-carbon compound. It's going to be used to produce CO2, and again, that CO2 is going to be used by rubip. Again, this rubip was something we had seen in the C3 plants. And that rubip is going to take that CO2, it's going to fix it, and it's going to continue with the normal process of the Calvin cycle. And we're still going to get the production of glucose as our byproduct. Remember, that's going to be the food for the plant. Alright, so that's going to finish up our second screencast for Chapter 10. It's going to be the final screencast for Chapter 10. Please remember to make sure that you have completed your screencast study guide.